Hi, I'm Savani. I'm an assistant professor in Adam Mickiewicz University in Poznan. And today I'm going to talk about one of the recent works on zebrafish ligand-receptor interactome atlas. And this work was led by a master thesis in bioinformatics student, Miłosz Chodkowski, and assistant professor Dr. Andrzej Zielazinski. And our inspiration for this work was my postdoctoral mentor, Gil Lefkowitz at the Weizmann Institute of Science in Israel. Our lab is interested in paracrine signaling and the formation of a small brain tissue called as the neurohypophysis, also known as posterior pituitary. So we are interested in the ligand-receptor interactions and how they promote tissue formation in the brain. And for this, we apply or we study using a model called as a zebrafish. It's a vertebrate model organism that has almost 70% of the genes conserved and similar to humans. The thing is, when we started our work back in, when I was trying to address this question back in 2016, I had the help of Dr. Dana Leshkovitz in the Weizmann Institute. And at that time, we did not have a specific tool for zebrafish. And so we were trying to make a very crude analysis based on other protein-protein interaction data available for human and mouse proteins, and all the omics data that were available only for mouse or human proteins and genes that are expressed in the tissue of our interest. But now, fast forward, and we are in 2023, we have single-cell sequencing, and we have all these amazing databases such as String. We even have specific customized tools that were built for human and mice data sets to analyze these data sets, the ligand-receptor interactomes, even Drosophila. So there are all these softwares called the CellPhoneDB, CellChatDB, FlyPhone software, and etc., etc. The thing is, zebrafish researchers like us, when we use softwares such as CellChat or CellPhoneDB, these softwares are basically built for human or mammalian data sets. And so when we will use such softwares, we will ignore zebrafish specific data. We will be only looking from a point of orthologous mammalian genes. So we will be forced to ignore either the interactome data of zebrafish or even the genes that are only in zebrafish and not in humans or mice. So this is a problem. And so what we decided was to first use one of the state-of-the-art algorithms called as DeepLock 2.0. And using this algorithm, we were able to predict the cellular, it's a machine learning algorithm. We were able to predict the cellular localization of the entire zebrafish proteome, because this is very, very important, especially because of the fact that we do not know the localization of a lot of zebrafish proteins. So for example, PIAS4A is a zebrafish protein which is known to be localized to nucleus, but then there is a protein Churchill, which we have, if you go to a UniProt data, nobody knows where is this protein to be localized. And that's why we need to use this latest machine learning algorithm, such as DeepLock, to predict where this protein can be localized. If we know if this protein is secreted, or it's bound on the extracellular matrix, or it is localized to the membrane, so then we could say, okay, maybe it is acting as a ligand, maybe it is acting as a receptor. And that's what we did. And once we sort of had this entire predictions, we also did some sort of manual curation to sort of narrow down a list of ligands and receptors. This is a very similar practice that has been also done by other creators of other softwares, such as cell-phone or cell-cell interaction database. And once we had this, we sort of made the entire software. We call it as a DanioTalk. We shared it. Our work is published in the iScience manuscript, and our software is open source. So it's in GitHub, DanioTalk. So I invite everyone to check our publication, see if it will be useful for you, and you could also use it for other model to make similar software and algorithms for other model organisms. And I thank you. So